Hey guys,
Hey.
it's your faves. Back at it again. Just kidding. Okay.
So we got Maddie, Tanner, and Liliana here, and also Braden, who's not able to do the video with us, but he did help us.
We had a family situation to handle, so.
So we give him credit for helping, but he is not present here today. But to get started, number one, we have a question dealing with genomic imprinting. In this case, it is the Angelman disease. So for A, it is asking if you cross a male, and a little bit more background. In this case, the male alleles are the ones that are silenced. So for A, the cross is with a heterozygous male and female. So we have a Punnett square right here. Am I pointing the right one?
Yes.
Okay, good. And in this case, we crossed out all alleles that came from the male because they are silenced and they aren't going to be counted in making this trait. And so the phenotypes that we get for this are half the offspring being normal and half of them diseased due to the male's alleles being silenced. For B, it's the same question, but you have a heterozygous male crossed with a homozygous recessive female. And so in this case, with the male's genes all being discounted, we get 100% of the offspring being diseased. For C, it is talking about bar bodies in X chromosomes. And for C, the situation is a woman with a triple X syndrome or having 3 X chromosomes. And in this case, the XICS, or the activation center, has been deleted from 2 of the X chromosomes. And it asks how many bar bodies it'll have. We put the answer as 0 because when the inactivation center is disabled, then none of the chromosomes will be inactivated, and in order for there to be a bar body, an X chromosome has to be inactivated. So there will be 0. And for D, it is a female with 2 X chromosomes, and the T6 gene on both of these is inactive. And since both of those are inactive, there's nothing to stop the XIST gene from inactivating both those X chromosomes. Therefore, both X chromosomes are inactivated, making it 2 bar bodies per cell.
So question number 2. So 2A, it's asking for an example of the holoandric gene, and we all agreed on SRY. B, it's asking for an example of a gene found on both X and Y chromosomes. And we said MIC2, and both of those answers were in the lecture notes or the professor notes. So we just based our answers off that. C, so C we drew it out right here, and the plus and minus is just to signify whether it's the IGF2 or the IGF2 minus. And so the question was asking if an IGF2 minus male mouse and an IGF2 minus female mouse crossed, what would the phenotypes of the offspring be? And we all agreed on a normal, and dwarf for the phenotypes. For D, the question, or it says, in the blank pattern of inheritance, an allele can be dominant in one sex while the same allele is recessive in the other sex. So this one would be sex influence, and that's related to one of the examples of male pattern, or just pattern baldness in males and females. For E, blank is an epigenetic process that occurs during gamete formation, so that one's genomic imprinting, while blank is an epigenetic process that occurs during embryogenesis, and that's dosage compensation. So basically it's just saying like genomic imprinting happens during this phase of the reproduction, whereas the dosage occurs in a different phase. For F, it says one of the topics discussed in lesson 2 involved the formation of a double-stranded RNA molecule. What two molecules form the two halves of the double-stranded RNA? So this kind of relates back to question number 1, and it's the XIST RNA and the T6 RNA. So they both act together and they create that double-stranded RNA. For G, it says when reciprocal crosses give identical results, the alleles that control the trait in question are located on autosomal chromosomes. And then when reciprocal crosses give different results, the alleles that control the trait in question are located on X chromosomes. And that one's just kind of self-explanatory, I believe. Number 3.
For question 3, it's about a woman's father has ornithine transcarbamylase deficiency, an X-linked recessive disorder producing mental deterioration if not properly treated. Can you make it a little better? Sorry, you're fine. If not properly treated.
I hate technology.
Sorry. The woman's mother is homozygous and is unaffected. And for letter A, what is the genotype of the woman's father and what is the genotype of the woman's mother? For 3, we will see that in this genotype, the father, he's affected, so he has a gene, and the mother does not have that allele present. When we have a cross, we're going to see that we're going to see that 50% of females are heterozygous but do not have OTD, and in males, 50% are unaffected. 50%.
They're just infection.
Okay. For B, the question is, if the woman has children with a man who does not have OTD, what is the chance that their first child will be a son with OTD? So there we're going to use the females from that first offspring, the woman, and we're going to cross that with a male who does have it. And so we're going to end up with a 1/4 chance that the son will have OTD with a genotype of X with the disease allele and Y. For C, the male would need to have genotype of, that one refers back to the question, identify the genotype of a male whom the woman could produce a daughter with OTD. There, the woman, we're using the woman from that offspring from the parents. She's heterozygous. The male would have to have that allele present. And the genotype that would daughters with OTD. So 1/4 of the daughters would have OTD, correct? Sorry. For D, so yeah, for C, the male would have to have X and the allele with the disease allele in order to have a daughter who has the disease. Sorry. And for D, when you do a cross, which basically would be that same cross, the phenotypes present would be 25% of them would be females, heterozygous for both alleles. 25% would be homozygous for the disease allele. 25% would be male with without the disease, and 25% would be also with the disease allele. And for four is a little bit easier for me to describe. Sorry, guys. The question is, the presence of beard in some species of goats is determined by an autosomal gene that is dominant in males and recessive in females. So for letter A, suppose that a cross produces 3 bearded, 1 no beard phenotype ratio among male offspring. What are the genotypes and phenotypes of the parents? So for there to be that phenotype of 3 bearded, 1 non-bearded in males, the homozygous dominant, which would be BB, that cross between male and female would have to be heterozygous for male and heterozygous for female, for there to be that the genotypes of homozygous dominant in males, heterozygous in males again, and then that 1/4 that is homozygous recessive, and that will give you the ratio of 3 to 1. For B, the question is, suppose that a cross produces a 1 beard and 3 no beard phenotype. In females, that capital B is recessive and that lowercase would be dominant. So in B, we're going to see that that ratio is produced when you cross the same male and female, heterozygous male and heterozygous female. You will get the same genotypes, but the phenotypes for the females will be different. In C, ratio among female offspring, what are the genotypes and phenotypes of the parents? That genotype would be homozygous dominant in males crossed with a heterozygous female who does not have a beard. And the phenotypes of the offspring would be 50% would be bearded females and 50% would be heterozygous, not bearded female. And for D, suppose a cross between a male with a beard and a female without a beard produces a 1 beard, no beard phenotype ratio among female offspring. What are the genotypes of the parents? For that one, C, I think I wrote it. Okay. So for that one, female offspring. Sorry, guys. I don't know if you guys can see it in my notes, but I have chicken scratch. Okay.
Sorry about this, guys. I had one job to write everything and I messed up.
Okay. Okay, I'm basically going to explain it, but it's you're going to have a cross between a homozygous male and a heterozygous female, and that's going to give us that ratio for D. And for is that it? Oh, E. E, suppose a male and a female, both which lack beards, produce a 1 beard. What are the genotypes of the parents? Actually, I didn't do E.
Explain this one. Okay, so for 4E, it says, suppose a male and female, both which lack beards, produce a one beard, one no beard ratio among the male offspring. What are the genotypes of the parents? So in this case, we know that the male has to be homozygous recessive, two little b's, because he lacks a beard, and the female also lacks a beard, but she can either be heterozygous or homozygous, little b's. But in order to produce a one beard, one no beard ratio among the male offspring, we know that the female has to be heterozygous, because as you do Punnett square, there has to be a dominant allele present in order for the males to be half bearded, half non-bearded. So that concludes 4. And moving on to 5, could you lower it? Thank you. All right, so this one is regarding cats and their coat color. The recessive allele in this case is orange. The dominant is black, and heterozygous, since it is incomplete dominance, is the tortoiseshell. So in A, it's asking, when you cross an orange male or recessive, homozygous recessive male, to a heterozygous female or tortoiseshell female cat, what are the genotypes and phenotypes of the progeny? Okay, so if we're looking right here, we see that as we cross these two, the male, and since this is an X-linked trait, the male has a recessive B on his X chromosome, and it's X-linked, so there's nothing on his Y. And then the female has a 1 dominant allele, 1 recessive allele. And that gives us the phenotypes of 1/4 being female tortoiseshell, 1/4 being female orange, 1/4 being male black, and 1/4 being male orange. For B, we have the question talking about it's asking, male tortoiseshell cats are very rare. What is the genotype of a male tortoiseshell cat? So in order to have a male tortoiseshell cat, he has to have an extra X chromosome. What's that called? Klinefelter's syndrome. And then he would have to be heterozygous on both of those X's. It's a very rare syndrome, and it means that he would also be sterile, but that is the only way that a male can be tortoiseshell. For C, it is asking, this one is regarding to fruit flies and whether or not they have normal margins on the wings or scalloped margins. And this one is also X-linked, and it wants us to do a what's called reciprocal cross to find out if this is, just to show that it is X linked trait that is linked to the sex chromosomes. So in this situation, let's see, the first one right here, the male is the recessive, homozygous recessive, and the female is homozygous dominant. And you do the cross, and we find that both the females and the males will all have normal, normal margins on their wings. And the second, when we do the reciprocal, where the male is homozygous dominant and the female is homozygous recessive, we see that there's a change, that the females are still normal, but the male offspring are all the mutant, the scalloped demonstration of that. And for D, it wants us to do the same thing, but for the autosomal chromosome. And in this case, we see that both sides are heterozygous in both instances, and that shows that this allele is linked to the sex chromosome and not the autosomal chromosomes. So that's all the problems. Thanks for watching.